Yeah, thanks everyone. So we're going to be taking some questions that have been asked over the last couple of days that we didn't get a chance to answer, as well as maybe some questions here live. So we've got a number of panelists here from a number of different disciplines here at PNI, and we look forward to some good conversation. So we'll start off with a question to Andy. Andy, do you see a future where traditional techniques such as live attenuated viruses or subunit vaccines are replaced by genetic vaccines?
Yeah, thanks for the question, Tim. I mean, we've seen COVID is the first example where we've really seen RNA vaccines competing head to head with conventional technologies, and we've seen the advantage. It's been rapid implementation of the technology, but it's a new technology, so we're not going to see it being introduced into pregnant women, potentially not into the very young, because there's no safety profile. The conventional technologies will use cell culture, but there's manufacturing infrastructure there. We're going to see these vaccines coming along a couple of months behind. There's every expectation that they're going to work too. And so we're going to see multiple vaccines implemented to try and contain the pandemic. Traditional vaccines that are already on the market are cost effective, they work, they're very expensive trials to do to show comparable efficacy. Hundreds of thousands of people. We're not going to see RNA technologies trying to displace those vaccines. It just doesn't make sense. Those vaccines are already licensed, they already work, there's already infrastructure in place to manufacture them. So we're going to see the new vaccine modalities. That's where we're going to see RNA being implemented. Particularly pandemic flu. We've seen challenges with Ebola, MERS, SARS, COVID-1 previously. That's where we're going to see RNA vaccines at the forefront. A lot's going to depend on the antigen, where the virus is coming from. If it's a bacteria, could be more difficult because expressing a bacterial antigen in a mammalian cell is problematic. And so there are going to be disease targets where RNA just isn't going to work. We're going to see, Robin Shattuck talked about, doing iterative antigen design for targets like HIV and TB. This is where RNA really is at the forefront, to do in silico design in an RNA format and then vaccinate humans to learn how the immune system can then adapt. We'll see a lot of that work happening in the future. We're seeing a huge investment by the Bill and Melinda Gates Foundation. We've seen Robin talk about his social enterprise, Vax Equity. We're going to see implementation of the RNA vaccine technologies in the developing world, trying to make cost-effective vaccines for new disease targets, where there's probably a niche market, small batches required, process of licensure is costly and time consuming, but there's money available through the foundations and non-profit companies to make that happen. So we're going to see a mixture of stuff happening in the future, but there will definitely, I don't think we're going to see it replace everything. I think it'll find a place where it fits, and there'll be a place where conventional technologies also fit.
Maybe as a follow-up question, Andy, a bit more specifically, what role could RNA play in things like seasonal flu, which are, halfway between continuously being developed and already approved?
Yeah, I mean, seasonal flu is well established in terms of production capacity. They just recently switched over from the swine flu in 2009. They really had a predominance of manufacture in eggs, which led to mutations and adaptations. There was the switch to cell flu culture. And bulk of the vaccines now are produced in mammalian cell culture for seasonal flu. The margins for those vaccines are quite small. Manufacturing's already established, and we're seeing quadrivalent flu vaccines being manufactured. Don't see RNA technologies displacing that traditional seasonal flu modality unless they find a way to do a universal flu, where they change the antigen design. Instead of expressing the full-length glycoprotein of hemagglutinin, they go with some kind of stalk where down the bottom you've got conserved protein structures, where there is a thought that you could raise a universal neutralizing antibody that would cover all the seasonal flu variants. That's where we may see a big paradigm shift. There's some data, it's not clear if that's going to translate to the future.
Okay, thanks. There was another question that was posed earlier during Peter's talk about, how crucial was the microfluidic mixing to the production of LNPs? And maybe this is a question more for that Lloyd can also chime in on, but the question was also about if you had taken a traditional method like thin film hydration, would it be possible to make something like Onpattro, and how different would that have been? And I think this applies to our LNP vaccines as well.
I was pioneering in the field for self-amplifying RNAs back with Novartis, and we would not have explored LNP delivery of a self-amplifying RNA if microfluidics hadn't existed, because we felt that other types of liposomal technologies were too fraught with scale-up issues. There's a graveyard of projects at the industrial scale that had just not made it through, and there just wasn't a willingness at the upper senior management level to see that even attempted. Microfluidics, we were allowed to take a look and see what it looked like. But Lloyd was embedded in Techmura during the days of Onpattro and pioneering that event, so I'll let him make a comment.
Yeah, I think the power of microfluidics, or PNI's microfluidics, is that it's open platform. Anyone can use it and get started very easily, whether they're developing a nanoparticle-based vaccine or a therapeutic. So I think everything that Andy said is completely on point. I think one of the biggest challenges is if you want to make a nanoparticle and you do not have a commercial suite of products available, you have to either read publications or patents and put it together yourself. And whatever you develop is going to be not exactly the same as the group in the patent or the group generating the publication. So that can take a long, long time to develop a robust and reproducible manufacturing process for nanoparticles. So there's a huge time saving to begin with. And we're seeing with microfluidics over a wide range of nanoparticle formulations, consistency. It's very predictable, the particle properties, the physicochemical properties that you're going to get. So when you take your expensive payload and the genetic medicine payloads, whether they're plasmid DNA, small interfering RNA, messenger RNA, or self-amplifying RNA, they're very expensive, and you want to ensure that you have a good process and you do not basically eat up a lot of this payload developing your product, because the costs become prohibitive very quickly. So you really want to be on point with your development activities, and you really want to do your process development work at small scale, so you don't have to do it at basically manufacturing scale, your PD work. And we're seeing very good, multiple projects, very good predictability of what we're going to get as we scale up. So, I think that's the power of microfluidics, that it's available. PNI's technology is widely available. We provide support all the way through, from discovery research all the way through clinical manufacturing, and hopefully beyond to commercial manufacturing. And we're here, we're available.
And certainly we have Peter Cullis, who's popped on the panel, so maybe he can talk about how microfluidics performed relative to conventional technologies in those early days as they converted over to it.
Yeah, I think particularly when you hit some formulations that are maybe a bit more difficult to formulate using some of the cruder approaches like T-junctions, etc. That's where the microfluidics really comes into play, when you have the very well-defined mixing process that's extraordinarily rapid. And so I think what Lloyd is saying is totally correct. that for those, particularly for the people that are not particularly used to doing formulation work, which is about 99% of the people that want to use lipid nanoparticles with mRNA or whatever inside them, it's a great advantage to have a technique that's rapid and reproducible and gives you well-defined product that you can then go and, as he says, not waste your precious mRNA or whatever other cargo you have, and so do this in a way that is going to be going to end up with something that you can actually rely on the results. I mean, one of the big problems with the whole field of nanomedicines has been this, you can understand in a way, but this urge that people have to go into in vivo situations with very poorly defined nanoparticles. And the result is, basically results that are irreproducible. And so the ability to go in with something that is a well-defined particle allows you to actually test how good your special ingredient might be, whether you're varying the mRNA or any other component. So it's really a vital part of this whole chain. And then, of course, the ability to move these things at the bench level to use exactly the same formulation process for a manufacturing process is a huge advantage.
Thanks, Peter. And maybe while we have Peter and Andy answering, this would be a good question for both of you as well. When you're thinking about a formulation for a vaccine, whether that be a lipid or a polymer, what are you looking for differently for a vaccine formulation versus a gene therapy formulation?
Was that question for?
Peter, if you could kick us off, that'd be great, and maybe Andy can add his thoughts afterwards.
Yeah, there are differences. The optimization for the vaccine point of view, I'm not as. I mean, it seems like the systems that work quite well in the liver work pretty well for vaccines. But the main point is you're not so concerned about stability in the sense of having a long circulation lifetime or those sorts of issues. Whereas, if you want to hit other tissues, as I mentioned a while ago, you want systems that are much more stable and they're able to maintain their integrity in biological fluids for some considerable length of time until they actually hit the tissues you want to affect. And so those are some real differences in formulation that we're going to see coming in more and more. Right now, we're liver-centric and vaccine-centric, for obvious reasons. There's some pretty big prizes or some pretty big applications there, but we are going to see other formulations come in that have different properties.
Yeah, I mean, I'll just comment. What we've seen is a lot of the liver-centric formulations being translated to use in vaccines, and they've worked very well. But the question you can ask, is it optimal? Probably not. What are you looking for in a vaccine setting? It's an intramuscular administration. You want it to remain local or traffic just to the draining lymph node, so it doesn't go systemic. I think Michael Houghton pointed it out, lipid nanoparticle with RNA inside, the immune system sees that as a viral particle. If it goes systemic through the draining lymph nodes and into the systemic circulation, you'll see a systemic antiviral response. And certainly that's what it looks like we're seeing, dose-dependent systemic type 1 interferon responses in the clinical trials. So I think we're going to see vaccines being optimized on the LNP side to be more local and less of it going systemic. And certainly, there was a nice elegant paper published by Moderna, showing how they'd modified the pKa of the ionizable lipid to maybe modulate that effect. And so we'll see. Maybe that's one way we'll see, the adverse event profile get better in the future for these vaccines.
Great, thanks, guys. And maybe shifting gears a little bit from the scientific, and Padma, I'll ask you this question first, but James, if you want to jump in, or anybody else, now, obviously, everybody is off to the races with a COVID vaccine. It's been a very impressive effort to get to where we are today. But looking towards the future, what are your thoughts, or what are you seeing in terms of efforts to expedite this even further for future scenarios?
I think the panelists did talk a little bit about it. Speed is important and supply chain becomes important. So the more groups that make available the mRNA, the key components, as we are talking about, you know, the mRNA, the lipid excipients, the components, the cationic ionizable lipid becomes important, the other lipid components, and then the manufacturing platform, obviously. So as these get more validated and more adopted into the field, you know, you'll just see speed and more and an easier way to get to these vaccines. So, you know, it's great with P&I being able to offer, you know, majority of this platform in an end-to-end solution that we do. And that is our hope, to be able to work with our clients to offer these.
Yeah, and just to add, I think this is benefiting not just the vaccine development, but it's validating the entire RNA platform in general. And we're going to see lots of continued growth in non-vaccine-based applications, I believe, because of these results and because of the impact it's making and the investments into RNA, LNP drugs in general. So it's exciting times for the field, for the entire field. And I think as we move forward, this class of drugs will continue to become bigger and bigger and more important, and accelerating with faster acceleration of these types of drugs. Great.
Well, I totally agree, James. It's this class of drugs is going to become. I mean, let's face it. If they're injected into a few billion people, they're pretty much mainstream now, or at least they will be by the middle of next year. You know, I mean, that's really, to me, just the tip of the iceberg. Yeah, because there's we can go in so many more directions, both for vaccine and therapeutic points of view. So it's an enormously exciting time. I've never seen anything like this, the way that this whole field is breaking open. So yeah,
very cool.
I think this is where everybody is just like, wow, 95% efficacy for two separate things. I mean, that really proves it out, that this approach on the vaccine side is absolute dynamite. But I mean, going back to the siRNA trial for Onpattro, I mean, look at the results there. When we start to go at things in a really fundamental way, using genetic drugs rather than small molecules, my God, I mean, this is transformative for sure.
Yeah, I think a cool thing that we see from our company and our perch of being able to work with so many different groups is we're seeing now these technologies go into the mainstream. So similar to what sequencers did for sequencing, and now every academic has a sequencer on their bench. I think the same concepts happening with these medicines, where you really can go from, once you understand a disease target, to a candidate that you can use to turn that target off or express the protein or edit it very, very quickly in a matter of weeks. And so the barriers being lowered dramatically, at least to get to the candidate level. And groups are, all over the map are coming into this field and have the ability. So hopefully has a big impact in increasing R&D productivity of pharmaceuticals and the like. It's tremendously exciting.
Yeah, I think one of the areas where you're going to see this is really going to press the regulatory authorities, but is in the area of rare disease, where you can pick up a problem from a hereditary point of view that somebody may have, and you can devise a medicine in the matter of a month or two, which then, hey, why not see if it actually works for the individual? And I think that's where there's huge opportunities in terms of getting approaches that allow us we don't change necessarily the carrier, we just change the cargo, and some relatively rapid way through the through the regulatory process to allow people that might have a huge need for it. We can make the liver make any protein we want right now. Well, just think about that. I mean, that means for blood clotting disorders, you name it, you suddenly have a therapy which wasn't available before that you can do really quickly. So I think that's going to be one of the transformative things as well, that we get used to a much more personalized medicine approach, simply because we can really focus in on the particular problem somebody has with great accuracy. It's going to pose issues from, as I said, regulatory and other points of view, but that's something that I think precision is ideally suited to start to exploit that. I think it's a huge opportunity.
Yeah, it's fantastic.
You know, I'll build on that. You know, we've seen the RNAi technology based on double-stranded siRNA. The drug, when it was first launched in the early 2000s, wasn't particularly potent. It really, you know, had off-target effects. We've seen the chemistry develop, and you know, now those GalNAc conjugates targeting the liver, they're dosing them twice a year and seeing, you know, 90+% knockdown of target genes. What we're going to see with the LNP field is new tissue targets being opened up. You know, the LNPs can be tissue centric. You've just got to look with the right analytical tools. So if, for instance, you find, you know, an LNP skewing towards the kidney, can you find a target in the kidney that's specific to the kidney that you can then tackle with an siRNA? And so even if the LNP is distributing, you know, to multiple tissue compartments, your drug is targeted specifically to the kidney, you have a therapeutic that you could then chase after. So, you know, we've talked a lot about mRNA, but let's not forget about, you know, knocking down genes as well. That drug class is exquisitely potent now, and it's ready to, you know, deliver to other tissue compartments.
Yeah, I totally agree. I mean, when we were working with Alnylam, of course, they were totally focused on sRNA and liver targets. I don't think they really dreamed that this kind of approach would work for mRNA. And of course, they became, the lipid nanoparticles. A bunch of chemists we're working with there. Chemists don't really like lipid nanoparticles. They want to have a small molecule that is well defined. And so there was two camps. You know, we were developing the nanoparticle stuff, which was working really well. But there was the other camp with the GalNAc, et cetera, saying, okay, well, maybe we can do this. But the thing is, they've ended up with something that's highly effective, but very limited. Whereas the lipid nanoparticle approach, okay, there's other tissues we can go after, other targets, other tissue targets, cell targets, et cetera. And not only that, we can put mRNA in there as well as it's. I'm sure they're kicking themselves a little bit, saying, "Wow, we sort of broke the barrier here for getting large molecules into cells, but not really realizing that you can get even bigger things in, which are totally. There's no way you're going to target an mRNA with a GalNAc or anything else. So the lipid nanoparticles, they're absolutely essential." And
I'd be interested in your thoughts, you know, Peter, around gene editing, because obviously there's, you know, single dose, potentially you can do a gene correction and in a rare disease have a huge impact. And it's early days, but, you know, you're already there in the liver. You've got probably more potent formulations now than MC3. We're several years on. And are you playing in that space with collaborations?
We're not at the moment. I mean, my focus has been more on, how do we get to extrahepatic tissues, and also just characterizing some of the, we put these lipid nanoparticles together. It's amazing how little we actually know about their structure. So I've been really focusing in on that the last while. But yes, I mean, you can see that, the gene editing approaches are on the table as far as the mRNA approach is concerned. And so yeah, I can, the writing's on the wall. I mean, I think this is going to be dominant.